Hi everyone, my name is Michela Deleidi. I'm a hallmarks investigator group leader at the German Center for Neurodegenerative Disease in Tübingen. And it's really a great pleasure for me to have the opportunity to be virtually back to the Neurodegeneration Institute at McLean Hospital, where I spent the years of my postdoc in the group of Ole Isaacson. So really a big thank to Ole, Oliver and Penny for this great opportunity. What I'm going to present you today is some of our recent data on the role of mitochondrial stress responses using human brain organoids. But before starting, really, I have to acknowledge all of my lab members in Tübingen. They've been really great, especially during these last few months. And concerning the project I'm going to present you today, I would really like to acknowledge two fantastic postdocs in my group, MJ and Dina. They really greatly contributed to the development of this project during the last few years and especially the last few months. So, in addition, of course, I would really like to thank and acknowledge all our collaborators, Massimo Zeviani, Carlo Viscomi, Dario Brunetti at MRC and U Cambridge, our local collaborators, in particular the group of Matthias Jukar with Stefan Kaiser and Rava, and at MPI in Tübingen, Katharina Hipp and Iris Koch. And last but not least, all the funding agency which made this project possible. So the topic is mitochondrial unfolded protein response. But what's a mitochondrial unfolded protein response? It's basically a mechanism which enables mitochondria to cope with the proteotoxic stress and to deal with protein folding and quality check in different stress conditions. Is based on a tight regulation and communication between mitochondria and the nucleus, which in case of a proteotoxic stress leads to the upregulation of chaperones, proteases, and different proteins involved in mitochondrial dynamics, leading to the upregulation, to the activation of different pathways, protective pathways for mitochondria, including antioxidant mechanisms, folding and degradation mechanisms, regulation of mitochondrial respiration and mitophagy, and many different other mechanisms. But what do we know about these mitochondrial stress responses? Definitely, we know from studies in simple organisms such as C. elegans, we know that this is a protective mechanism. Upregulation of mitochondrial UPR is beneficial, increases lifespan. And we also know that mitochondrial stress responses are upregulated in the course of aging and also different diseases, including brain diseases. Different genetic mutations and also environmental insults can indeed trigger mitochondrial stress responses. However, we don't know whether these mechanisms, these mitochondrial stress responses, are actually a protective mechanism or detrimental. And this is very important because it can also give us information about the pharmacological targeting of this mechanism. So this is the central question of this project. We wanted to dissect the role of mitochondrial UPR in brain disease. And to do that, we basically based our study on a model of a primary mitochondrial disease linked to mutations, loss of function mutations in a gene called PTRM1. The groups of Massimo Zeviani and Loris Bindorf have identified families carrying loss of function mutations in this gene, and these patients develop a childhood onset cerebellar disease with ataxia, and also a slowly progressive syndrome characterized by a mental retardation, psychotic symptoms, and cognitive decline. So the progression of these symptoms is really slow till their late 60s, And this is quite unusual for common mitochondrial diseases. So to further understand the role of these peptidase, as I said, PTRM1 is a mitochondrial peptidase. The groups of Massimo Zeviani have also developed an experimental model system based on heterozygous PTRM1 knockout mice. It's important to note that the knockout of PTRM1 is embryonically lethal. In heterozygous knockout mice, what we can see is the development with the aging of brain pathology which resembles AD pathology, with the presence of A-beta deposits. But what's the link between these mitochondrial peptidase and Alzheimer's disease and A-beta metabolism? So PTRM1 is involved in the degradation of the mitochondrial targeting sequences of the proteins that are imported into mitochondria. And original studies conducted in East have shown that the deficiency of the function of these peptidase leads to the accumulation of mitochondrial targeting sequences, which are toxic and leads to mitochondrial dysfunction with a reduction of mitochondrial membrane potential, reduction of oxygen consumption, and also some increase of reactive oxygen species. But where is the role of beta and a possible link with Alzheimer's disease in this pathway? So it's important to remember that several studies in the last years have shown that amyloid beta is imported into mitochondria. This is an EM study of AD brain compared to a non-affected brain, and we see that we have indeed accumulation of amyloid beta within the mitochondria. And also, we know that it has been shown that this mitochondrial peptidase is also involved in the degradation of the mitochondrial portion of A-beta. So a loss of function of PTRM1 leads in vitro to accumulation of A-beta within the mitochondria. And A-beta, in terms with a negative feedback loop, can inhibit PTRM1. So this is a potential link to what the role of PTRM1 and what observed in these patients. As I said, these patients develop cognitive declines, which with a slowly progression, which is quite unusual for this disease. As I mentioned before, the knockout of PTRM1 is embryonically lethal. So we wanted to look at the function of PTRM1, and to do so, we knocked out PTRM1 in human-induced pluripotent stem cells using CRISPR-Cas9 technology. We have differentiated induced pluripotent stem cells into cortical neurons. And first, we have looked at the basic function of mitochondria. So we looked at different aspects, including oxygen consumption, mitocyte production, mitochondrial membrane potential, and several others. In general, what we have found is that in cortical neurons generated from PTRM1 knockout-induced pluripotent stem cells, mitochondrial function is quite conserved. And what we could observe is only a significant reduction of mitochondrial membrane potential in PTRM1 knockout cells, but only at the level of the neurites. So again, something which could mimic what really happens in Alzheimer's disease patients at early stages. Indeed, we also found that these mitochondria are more ubiquitinated, and also when we looked at the mitochondrial content, we saw a significant reduction of mitochondrial content in PTRM1 knockout neurons. So these data suggest indeed an increased mitochondrial clearance in PTRM1 knockout cortical neurons. We are interested, as I mentioned, in mitochondrial stress responses. PTRM1 is not directly involved in mitochondrial proteostasis. However, as I mentioned, dysfunction of these peptidase can indirectly link to defects in mitochondrial function. And what we found, we checked both the gene and the protein levels, the levels of different proteases and chaperones involved in mitochondrial stress responses, and we indeed found a significant increase of mitochondrial stress responses linked genes in PTRM1 knockout neurons compared to wild type. So we have an induction of mitochondrial stress responses. Since we know that mitochondria, it's known that stress responses within mitochondria may also impact quality control mechanisms in the cytosol, we looked at autophagy, and we found that PTRM1 knockout neurons have an increased autophagic flux. And in addition, we also found an increase of ubiquitination in PTRM1 knockout neurons, which is important for some data and findings that I will show you in a minute. As I said, PTRM1 is linked to the degradation of the intramitochondrial portion of A beta in vitro studies. At least this has been shown. What we found is an increase of duration A beta 140 to 140 in PTRM1 knockout neurons, along with an increase of amyloid precursor proteins. So this was also accompanied by a decrease of the mRNA levels of APP. So we don't have an increased synthesis of APP. So this data, along with the data that I've shown you before related to the ubiquitin levels, the total ubiquitination in knockout neurons, suggest that there is also a defect in quality control mechanisms, and in particularly, it's likely that these mitochondrial stress responses lead to an overload of the proteasome. So what we found in cortical neurons that we have generated from induced pluripotent stem cells, wild type, and PTRM1 knockout neurons, we found that deficiency in the function of mitochondrial UPR leads to upregulation of activation of mitochondrial stress responses, in increasing mitophagy, deficiency, defect in quality control mechanisms at the level of the cytosol, with accumulation of APP and increase of A beta. However, if we look at the main pathological hallmarks of Alzheimer's disease, namely AB plaques, tau pathology, and cell death, we couldn't find any of these pathological hallmarks in our models. So I'm not showing the data, basically it's a list of negative results, but we didn't detect any of these hallmarks. So we then decided to turn into a more complex model, so namely cerebral organoids. And for all these part of the project, I would really like to thank Maria Jose Perez. So MJ is a fantastic postdoc in my group. She's a EMBO fellow, and she really developed all these part of the project based on human cerebral organoids. So human cerebral organoids, on one side, they give us the opportunity to work with long-term cultures. So we were able to keep these organoids in culture up to 6 months, and more recently up to 1 year. And these provide a better environment for neuronal maturation. But also, one of the advantages is the presence of different cell types. So they allow the analysis of cell-cell interaction and also cell type specific mechanisms in the context of different diseases. What we did is to generate PTRM1 cerebral organoids, both wild type and knockout. We didn't see any defect in the growth and in the survival, the overall survival of these organoids in culture. So what we observed when we looked at the cerebral organoids, these are organoids from wild type and these are PTRM1 knockout organoids, is that we found an accumulation of ubiquitinated proteins. This is at approximately at 65 days of growth. We also found a protein aggregation, so A-beta-like plaques here with a BTA1 staining in PTRM1 knockout cerebral organoids, along with tau pathology, increasing tau phosphorylation, and cell death as revealed by an immunostaining for cleaved caspase 3. So using this complex model, we were able to observe AD-like pathology in cerebral organoids. As I said, one of the advantages of cerebral organoids is that they contain several cell types which may better reflect the complexity of the human brain, both during development and disease. What we did is to run a 10x genomic study on cerebral organoids, so to analyze at single cell resolution the composition of our cerebral organoids, both wild type and PTRM1 knockout. These are all the cell clusters we were able to identify in our cerebral organoids. We identified 25 clusters that we further grouped in 4 major subtypes: astrocytes, microglia, neural stem cells, and the cortical neurons. And these are some of the genes that we employed to cluster our cell types in cerebral organoids. When we looked at the differentially expressed genes in our clusters, what we found is that in cortical neurons, astrocytes, and microglia, so these are the differentially expressed genes and the pathways that are dysregulated in the different cell types, we found that cortical neurons, astrocytes, and microglia display actually as the most dysregulated pathway mitochondria, so including oxidative phosphorylation and mitochondrial dysfunction, something that we didn't came up out in the neural stem cell cluster. So, and this is likely reflected by the fact that we know that from the neurogenesis studies that the neural stem cells are most glycolytic. It's also important to see that we were able to detect the dysregulation of immune pathways in astrocytes, and is something that MJ is currently working on. And also concerning microglia, so these cerebral organoids contain a really low proportion of microglial cells. There are some microglia-like cells. However, in these cell clusters, as I mentioned, we could detect oxidative phosphorylation and mitochondrial dysfunction, a dysregulation of these pathways. And when we looked at specific genes, it's very interesting to note that we could find an upregulation of the genes that have been described in the literature as early microglial genes in studies from single-cell studies from Alzheimer's disease patients. So this model may resemble also from an immune point of view early stages of Alzheimer's disease pathology. As I said, we are interested in a particular aspect of mitochondrial function, in particular in mitochondrial stress responses. Again, we analyzed the different chaperones and different proteases involved in mitochondrial stress responses. And similarly to what observed in 2D models, we could observe, we detected a significant upregulation of these genes. So we see an upregulation of mitochondrial stress responses also in cerebral organoids. But what's the role of mitochondrial stress response in this context, in this disease model? So to dissect the role of mitochondrial UPR, we use different pharmacological approaches. So first, we employed an inhibitor of the integrated stress response, ISRIB. This is a potent inhibitor of all the signaling of the integrated stress response. So organoids were treated with ISRIB. And what we could detect is that if we inhibit the integrated stress response, we have an increase of cell death, also an increase of tau pathology, of tau phosphorylation, along with an increase of APP levels and A-beta 42-140 ratio. So these data suggest that the stress responses that we have observed in our model system are actually protective. To confirm this hypothesis, we used another pharmacological approach. So we wanted to trigger mitochondrial stress responses. What we employed is an NAD precursor, nicotinamide mononucleotide. We have previously used NMN as an AD booster in cell models of Parkinson's disease. And we have seen that actually NMN is efficient in triggering mitochondrial UPR in IPS model systems. So what we did here is to use NMN to trigger, to further enhance mitochondrial stress responses in brain organoids. And interestingly, we could see a rescue in terms of cell death. So we could see that the cell death is reduced upon treatment with NMN. And we could also observe a reduction of the A-beta for 140 to 140 ratio. Importantly, we also see that NMN induces mitophagy. So we check the mitochondrial content. Here are our PTRM1 knockout cerebral organoids, and we could see a significant reduction of mitochondrial content, which suggests enhancement of mitophagy. It's also interesting to note that, however, we have an opposite effect in wild-type organoids. So in the wild-type setting, we have an enhancement of mitochondrial biogenesis. So here we have, in fact, an increase in mitochondrial content. And we are actually further dissecting the mechanisms underlying these different action of NMN in the healthy and disease condition. So, as a summary, we have dissected the role of mitochondrial stress responses using both cortical neurons and cerebral organoids from PTRM1 knockout-induced pluripotent stem cells. We have analyzed the role of these mitochondrial peptidase, and what we have found is that in our model systems, we don't see an accumulation of A-beta within the mitochondria. What we have observed, what we have found is a strong upregulation of mitochondrial UPR, which leads on one side to protective mechanisms, and on the other side also may interfere with the cytosolic proteostasis, leading to a beta pathology. So it can be that the induction of mitochondrial UPR over time is not sufficient to counteract these defects, these detrimental mechanisms occurring within the cell. We have also demonstrated the protective role of mitochondrial UPR in our model systems. Indeed, using NID boosters, as I said, NID boosters including NMN are able to induce mitochondrial UPR. We have observed that NMN leads to a reduction of A beta pathology along with a rescue of tau pathology and cell death. And we are now further investigating on one side the role of the chronic activation of mitochondrial UPR in the context of brain disease, and on the other side the possible pharmacological interventions that could rescue the disease-related pathological hallmarks. And with this, a big thank, as I said, to Ole, Penny, and Oliver for this great opportunity.